All right, excellent. Thank you so much, David, for being here, and thank you everyone for joining us. Welcome to the 2026 AGM Preclinical Translational Science webinar series. For who that doesn't know me, I am Serene Markosian. I lead the Assay Guidance Manual program here at the National Center for Advancing Translational Sciences, which is part of the National Institutes of Health. I'm super excited today to introduce our speaker, who's Professor David Lear. David is the Richard Merkin Professor and core faculty member at the Broad Institute of MIT and Harvard, the Thomas Dudley Cabot Professor of the Natural Sciences at Harvard University, and also a Howard Hughes Medical Institute investigator. David's research integrates chemistry and evolution to eliminate biology and enable novel therapeutics. Technologies that have been pioneered in his laboratory include base editing, prime editing, phage-assisted continuous evolution, PACE, and DNA templated synthesis. These technologies are used by thousands of labs around the world to study and treat genetic diseases, and at least 23 clinical trials using base editing or prime editing are underway. 8 trials are actually reporting results to date, which are yielding to medical milestones, including the first treatment of a patient with base editing, the first treatment of a patient with prime editing, and the first correction of a pathogenic mutation in a human. He is an elected member of the U.S. National Academy of Science, National Academy of Medicine, and American Association for the Advancement of Science, and he has been recognized with distinctions, including the 2025 Breakthrough Prize in the Life Sciences, the 2025 FNIH Montrone-Seigel Prize, and the 2026 Harvey Prize. He's also a founder or co-founder of biotechnology companies, including Prime Medicine, Beam Therapeutics, and Editas Medicine. Today, David's talk is entitled Mutation-Specific, Mutation-Agnostic, and Disease-Agnostic Therapeutic Genome Editing. If you have any questions for David during, please place the questions in the Q&A box, and we will get to them at the end of the presentation. David, on behalf of NCATS and all of our attendees, we're super excited to have you here today. The floor is yours.
All right. Thank you very much. It's an honor to present to this group. I have long appreciated that NCATS is sort of the tip of the sphere of our country's translational research and medicine efforts, and hopefully this talk will be relevant to some of the ongoing interests of NCATS. So today, as the title implies, I'd like to describe 3 distinct strategies for therapeutic gene editing. And these strategies each illustrate how the development of gene editing technologies enables us to pursue a variety of therapeutic approaches, some of which have already had a substantial impact on patients. But I think the sort of meta theme of my talk is that scientific advances and their creative application can also help address some bottlenecks that are traditionally considered to be non-scientific, practical bottlenecks, regulatory bottlenecks, economic challenges. And those are really many of the bottlenecks that stand between connecting the science of gene editing with patients currently. Okay, so I think this audience is probably very familiar with the problem that our lab is trying to address. And that problem is that there are a large number of people, by some estimates around 400 million, that live with genetic disease. Far more patients live with genetic disease collectively than live with cancer or with HIV/AIDS. And yet, if you examine the investment globally per patient, you can see that rare genetic disease patients, despite being so large and many suffering from severe diseases such that half of these 400 million patients are children and 30% of that half will die before their 5th birthday because of their genetic disease. So 60 million kids currently alive are predicted to not make it past their 5th birthday because of their genetic disease. Yet global investment in rare genetic disease research is about $3 per patient. So that's really the problem that I'd like to present today, which is there are thousands of diseases, hundreds of thousands of pathogenic mutations that cause these diseases, yet most mutations occur in a number of patients far too small to be considered, at least by traditional biotech metrics, commercially viable. And the question that this talk will try to make progress on addressing is, how can many small patient cohorts be unified so that a single treatment can benefit as many patients as possible? Okay, and so that's really where these three strategies come in. The first strategy of therapeutic gene editing is one that everybody, I think, is already familiar with, mutation-specific strategies. If there's a mutation that is known to have a very high penetrance, to have a very high probability of causing a genetic disease, then in theory, we should be able to correct that specific pathogenic variant and perhaps benefit patients. But that approach is commercially viable for only a small handful of genetic diseases under the current regulatory and commercial environment. And to be clear, I am optimistic, and I think all of you have heard FDA and other countries' regulatory agencies making progress about recognizing the enormous need in this space and the urgency of streamlining those regulatory requirements so that the process of gene editing becomes regulated much more in a process-oriented platform way instead of a composition of matter way. All of that, I think, is inevitable. It is coming, but it will take time. And in the meantime, we can consider other approaches to therapeutic gene editing that can complement the mutation-specific approach and help us address this problem. So I'll talk about a mutation-agnostic approach, for example, integrating a healthy gene copy at a target of endogenous gene loss or mutation. And then finally, I'll talk about a disease-agnostic therapeutic genome editing approach. That is, how can we use gene editing to remedy common information flow errors in biology that underlie not just one disease, but multiple different diseases. All three of these examples have some useful specific case studies. I'll use, one of the actually not the first, one of the last, most recent base editing or prime editing clinical trials is that of KJ Muldoon, but one that I think all of you have heard about in the news. This is a great example to exemplify all three of these strategies because baby KJ inherited two mutations, one from dad and one from mom, in his CPS1 gene. The mutation from dad is shown here. That's the one that we corrected in an effort led by Kiran Musunuru and Rebecca Ahrens-Nicholas. That's a mutation-specific therapeutic gene editing strategy. Use an adenine base editor to correct this T back into a C. N of 1. There's only one patient known to have this particular mutation, and that's baby KJ. Sorry, one patient with CPS1 deficiency, meaning with two bad CPS1 copies. KJ's dad, of course, has the mutation as well. The second strategy can also be applied to CPS1 deficiency because if you do an analysis of how many loss-of-function mutations afflict patients, loss-of-function mutations in CPS1, and imagine a therapeutic that would integrate a healthy copy of CPS1 at the right environment in the genome to ensure its proper regulation and expression level, the answer is something like 1,000. Maybe 100 new patients per year have CPS1 loss-of-function biallelic mutations, or two bad copies. And then finally, if you think about this case in a broader sense, this is an example of a very common class of mutations that transcends many different organs, many different diseases, many different symptoms, and that is it's a premature stop codon. a CAG glutamine codon has been changed to a TAG premature stop codon. Perhaps we could use gene editing to rescue many or all TAG premature stop codons. And if you do an analysis of how many patients suffer from, we'll limit it to liver diseases caused by premature TAG stop codons, you come up with an answer that's about 1 million. So these three strategies differ by 3 orders of magnitude in each successive tier. And that sort of exemplifies the kind of strategies that our lab thinks a lot about, how to amplify the patient impact by trying to creatively use gene editing and develop new kinds of gene editing. Okay, so we'll start with mutation-specific gene editing, specifically base editing and prime editing. There are only 3 ways that humans have edited the genome of cells in patients. You can use a nuclease to cut DNA, you can use a base editor or a prime editor. Those are the only 3 ways that humans have edited human DNA in the clinic. The latter 2 are really technologies our lab developed to precisely convert a sequence of your choosing into a different sequence of your choosing. So these are engineered proteins. They don't exist in nature. They make specified changes in DNA without requiring double-strand DNA breaks or donor DNA templates. And base editors make these kinds of changes. Prime editors can make virtually any kind of local change. And our lab and many others have used base editors and prime editors, both ex vivo and in vivo, to treat animal models of genetic diseases. Shown here are some of our earliest examples where we used base editing to rescue sickle cell disease in mice, ex vivo, and in this case, in vivo base editing to rescue progeria in mice. And in both cases, we observed extensive rescue of disease phenotype. And there are now many, many examples from many labs of using these technologies to rescue animal diseases, animal models of serious human genetic diseases. An example of a prime editing therapeutic application to rescue an animal model of a serious genetic disease is very recent work we published in collaboration with the labs of Kat Lutz and Al George. This was the use of in vivo prime editing to rescue a disease called AHC, alternating hemiplegia of childhood. It's a severe neurodevelopmental disorder that presents with paralysis, involuntary movement, and seizures. And there's no effective treatment. We developed base editing and prime editing strategies that correct with roughly 40 to 90% efficiency 5 ATP1A3 mutations that collectively cause about 70% of the known ATP1A3 associated AHC cases. One interesting footnote is, although 4 of the 5 mutations are transition mutations, and therefore in theory can be corrected by base editing, we ended up choosing prime editing to correct 3 of these 4 transition mutations because of the presence of non-silent bystander edits that resulted from base editing these alleles. But in every case, even though we went in not trying to use base editing or prime editing in particular, we were able to find either a prime edit or a base edit solution that could efficiently correct these mutations in human cells. And then to skip a lot of preclinical work, and jump to the in vivo work, we used AAV9 mediated in vivo delivery of an optimized prime editing system to prime edit the CNS of 2 AHC mouse models. And we were actually quite happy with the efficiency we observed. We obtained about 30 to 50% bulk brain cortex correction, and among transduced cells, about 50 to 90% correction. And then if you look at the mRNA level, we see correction even at higher levels, suggesting that prime editing is occurring efficiently among the cells that express ATP1A3, likely reflecting the neurotropism of AAV9. And then importantly, the treated animals showed substantial restoration of hippocampal ATPase activity of ATP1A3, which is the main activity that's missing in patients with this disease. And then both prime editor injected mouse models, both of these different mouse models that differ in their disease severity and presentation. They both showed strong amelioration of stress-induced seizures, movement and cognition deficits, and quite pronounced extension of animal lifespan. And so this work is an example of how it may be feasible to use prime editing as a potential one-time treatment for AHC. And to our knowledge, also represented the first use of prime editing to rescue a neurological disease in animals. And we're currently working with lots of kind souls to try to advance this work through IND-enabling studies into a clinical trial. All right, so therapeutic editing results such as these, along with the contributions of many, many other labs that are now described in more than 2,500 base editing and prime editing papers to date, have led to at least 23 ongoing clinical trials using these technologies, as was mentioned in the introduction, to treat a wide range of diseases, both ex vivo and in vivo. In fact, you might be surprised to look at this chart and realize that the majority of these clinical trials are actually in vivo, not ex vivo. The diseases include a variety of blood disorders, an immune deficiency, heart disease, metabolic diseases, amyloid disorders, muscular dystrophy, even a neurodevelopmental disorder, and others. And now 9 of these clinical trials have reported results because one read out just a week or two ago, this one from Yoltech. And in all 9 cases, happily, the base edit or the prime edit has been shown to benefit patients. So I'll now just highlight some of the, I think, key milestones in these clinical trials by summarizing a few of them. So this is Alyssa Tapley. At the time she was treated, she was a 13-year-old girl in the UK suffering from refractory T-cell leukemia. And in 2022, she became the first patient treated with a base-edited therapeutic when she received CAR T-cells in Wasim Qasim's trial. These CAR T-cells contained 3 base edits that enabled them to attack her cancer, but not each other and not her healthy cells. And her T-cell leukemia went into complete remission, and she's now more than 3 years after treatment. Her cancer remains undetected. She's back in school. She hopes to pursue a career in biomedicine, and I had the great pleasure of meeting her last April. This is Brandon Baptiste. He suffered from severe pain crises, replacement of both hips, and acute chest syndrome caused by blocked blood vessels in his lungs because he suffered from sickle cell disease. He received an autologous transplant of his own hematopoietic stem cells that were base edited in the fetal hemoglobin promoters to reawaken fetal hemoglobin expression. And the result is that Brandon is now free of pain, pain crises, and acute chest syndrome, and is no longer receiving any medication for his sickle cell disease. In late 2023, the first clinical trial results were announced from the first in vivo base editing trial. This is a collaboration between Verve Therapeutics and Beam Therapeutics. Verve was, of course, then purchased by Lilly. But patients with very high levels of LDL cholesterol were given a single injection of an adenine base editor programmed to precisely disable a splice site in PCSK9. And the result is that LDL cholesterol levels in these patients were durably, probably permanently lowered by up to about 60%. So this is an interesting case because it demonstrates the potential of in vivo base editing to not just treat serious disease, but really to lower future disease risk, in this case by introducing a precise substitution into a wild-type healthy copy of a gene that all of us have. So this is data from a trial that read out in March of last year, and I think it really represented a milestone in medicine. This is the first correction of a pathogenic mutation in a human. So this is a work led by scientists at Beam Therapeutics. They used an LNP-delivered adenine base editor to correct the most common cause of alpha-1 antitrypsin deficiency. This is a lung and liver disease affecting 100,000 people in the US alone. So it's a fairly common disease as rare genetic diseases go. All right, and as many of you saw this past May, a team, again, led by Kiran Musunuru and Rebecca Ahrens-Nicholas, achieved, I think, another medical milestone. This is the first use of a bespoke gene editing therapy to treat a mutation found in a single patient. So this is baby KJ Muldoon, who, as I already mentioned, suffered from a urea cycle disorder. This type of disorder has about a 50% infant mortality rate, and in KJ's case, was caused by a compound heterozygous mutation in CPS1. And so in a remarkable testament to the dedication of the team and regulators, manufacturers, and the robustness of the underlying science, an adenine base editor was developed in about 6 months and dosed less than 7 months after diagnosis. This is a process that you would normally associate with a much longer multi-year effort rather than 7 months. But the result is that baby KJ is now thriving. And I think this example really served to bring to the public consciousness that we do have the ability now to go into a person, fix an otherwise fatal mutation, and in some cases, the patient can actually begin to thrive and hopefully live a fairly normal life. Recently, the results of the first prime editing clinical trial were read out. This trial began only 4.5 years after we published the very first prime editing paper in late 2019. The disease is chronic granulomatous disease, CGD, and it's caused by mutations in NADPH oxidase subunits. Loss of 2 base pairs, GT, in a gene called NCF1 is one of the most common causes of CGD. And a prime editing system that corrects this mutation by reinserting the missing GT can very efficiently not just correct the mutation and do so without affecting engraftment of long-term hematopoietic stem cells, but also rescue the previously missing enzyme activity and edits with no detected off-target edits in human hematopoietic stem cells. And then that paved the way for this trial where 2 patients have been treated thus far, an 18-year-old and a 56-year-old. I think it's fair to say the results of both patients that have now been shared has been beyond what anybody could have hoped for. They experienced rapid, so this is ex vivo electroporation of their hematopoietic stem cells with prime editor messenger RNA programmed to correct the missing GT, reinsert the GT exactly at the right place in NCF1. The procedure was very gentle to their HSCs, as evidenced by rapid engraftment by day 14 and differentiation to the normal subsequent blood cell lineages ahead of schedule, restoration of NADPH oxidase activity in a large fraction of these neutrophils. Editing was, in the 60 to 80% range, so quite efficient. And both patients have already experienced clinical benefit already. They have both observed amelioration of inflammatory symptoms of this disease. You can go on social media and see a picture of the 18-year-old snowboarding now. And the results have really, as Tracy Atteberry, the second patient, noted, the results have been life-changing. In fact, Tracy's brother died when he was 3 years old from the same disease. Okay, so those are all examples of mutation-specific therapeutic gene editing. And to be clear, I think that's a very important activity. It's arguably the most de-risked from a biological point of view when the genetics are already very well established. But an approach we became quite interested in to develop mutation-agnostic therapeutic gene editing is the one I'll talk about next, integrating a healthy gene copy at a target locus. This is, of course, not a new idea. It's a decades-old idea, first reduced to clinical practice 35 years ago or so with the development of SCID gene therapies to treat what used to be called bubble boy syndrome. In theory, this approach might benefit any patient suffering from a loss-of-function genetic disease, regardless of that patient's specific mutation. But I think one of the lessons of what I'll call gene therapy 1.0 is that it can be beneficial, it can be important to introduce the healthy gene copy at a targeted location in the genome of the patient, such as its endogenous genomic locus, to preserve as much of the native regulatory context of the gene, or at a safe harbor locus where its expression is less likely to interfere with other essential genes. That's preferable, I think, to just overexpressing unregulated amounts of a gene product. So our lab has developed two approaches to achieving the goal of targeted, that is programmable, gene size integration in mammalian cells. One approach is to use prime editing to install a recombinase landing site at a target location, and then use a recombinase to integrate the cargo gene into the prime edited landing site. We evolved a BXB1 recombinase that is more efficient than the wild type recombinase at doing this. And using this approach, which we call passage, we can efficiently install therapeutic cDNAs into disease-relevant target genomic sites in human and mouse cells. And we're currently in the process of advancing this technology into animal models as well. A second method to achieve targeted gene integration is the result of a longstanding collaboration with Sam Sternberg's lab to evolve CRISPR-associated transposases, or Cas, to work efficiently in mammalian cells. Cas are remarkable molecular machines. They perform in bacteria very efficient large gene, tens of kilobases, integration with very high specificity. They can be retargeted in a guide RNA programmed manner. They don't make free double-stranded breaks as part of their mechanism, and so they don't generate large excesses of indels. In fact, we don't really detect any indel formation from most Cas, and their product tends to be fairly high purity, meaning there aren't a lot of byproducts. The main downside of CRISPR-associated transposases for therapeutic uses has been that in mammalian cells, they typically show very low activity. And in fact, if you take a broad harvest of, in this case, type 1 Cas homologs that Sam's lab did, they found that the best of these homologs support maybe 0.1% targeted gene integration in the genome of mammalian cells. It's tempting to speculate that unlike the bacterial adaptive immune systems like CRISPR nucleases, Cas probably didn't evolve to be maximally active. And so we speculated that maybe transposition catalysis or DNA binding might limit integration in mammalian cells. But since the molecular determinants of those properties aren't really known in Cas, we thought this is a challenge, or the challenge of developing Cas variants that can function efficiently in mammalian cells is ideally suited to protein evolution. And so in work that was initiated by former graduate student Shannon Miller and co-led by Isaac and Simon in our lab and by George and Sam Sternberg's lab, we used PACE to evolve CRISPR-associated transposases through thousands of generations of mutation, selection, and replication. One of the selection circuits we used is shown here. In the interest of time, I won't go through it. But I'll just say that integration ends up placing a promoter in front of an otherwise promoterless gene that's essential for phage propagation. And that's the basis of how we could link Cas activity to phage propagation in PACE. And after using a number of different PACE circuits and campaigns to work on this problem, we achieved, through this campaign shown here, some highly evolved TNSV variants like this one that ended up as the product of hundreds of generations of mutation, selection, and replication, which would have probably taken us decades, literally, to perform using traditional protein evolution methods. These mutations emerged in lots of different regions of the Cas components, but many of the most important mutations are predicted to lie at the interface of TNSB, which is the catalytic subunit of the transposase, and other components of the Cas complex. And when we reverted each of these mutations in TNSB, we found that all of them contribute to improved Cas activity, and some of them, very strongly so. So when we assess the ability of these PACE-evolved Cas to integrate large DNA donors into targeted genomic sites in human cells, we were really excited to find that while wild-type Cas mediated less than 0.1% genomic DNA integration, in this case in HEC293 T cells, the PACE-evolved Cas mediated guide RNA program DNA integration several hundredfold more efficiently. The PACE evolved P4-15 variant, which came right out of the final PACE lagoon, could integrate DNA cargo into 3 genomic test sites in human cells with an efficiency of roughly 10 to 20%, without any enrichment for transfected or edited cells. The contribution of all the evolved mutations in this variant greatly improved mammalian cell activity, and so we began to wonder what the basis of that was. And based on their location of the mutations in the predicted structure of the transposase DNA complex, our hypothesis is that PACE actually enhanced multiple functions in TNSB, and indeed we found that PACE improved transposase binding of the transposon ends in human cells, as well as integration kinetics of the evolved transposase, even in just E. coli cells. And then to determine if EvoCast can function efficiently at therapeutically relevant applications, we tested their ability to integrate donor DNA relevant to the treatment of genetic diseases in a dozen genomic sites of clinical interest, including 4 safe harbor loci, the track locus for CAR-T engineering, and 7 genomic loci where loss-of-function mutations cause Stargardt disease or cystic fibrosis or Fanconi anemia or sickle cell disease, beta thalassemia, SCID, Rett syndrome, or phenylketonuria. And once again, we generally observe DNA integration efficiencies of, say, 10 to 30% in HEC cells, with an average efficiency of 13% at these therapeutically relevant loci. And comparing the efficiency of the evolved Cas to the wild-type Cas at these 13 therapeutic sites, again, revealed an average improvement exceeding 350-fold. And so while I think we can still improve the efficiency of EvoCast, and we have a number of ongoing efforts that are bearing fruit in that regard, its integration efficiencies are already sufficient that if we can translate them into this level of integration in relevant cell types in a patient, they would offer potential benefit to some patients suffering from a variety of loss-of-function genetic disorders. Finally, we extensively characterized the integration products from our evolved Cas and found that the evolved Cas retains many of the favorable attributes of the natural type 1F Cas system. EvoCast is quite regioselective for cargo installation, which occurs predominantly 48 or 49 base pairs downstream of the target sequence. It's also predominantly unidirectional, just like the wild-type Cas, with over 90% of insertion events occurring in one orientation. And again, consistent with the fact that the mechanism of Cas proceed without the formation of free genomic double-strand breaks, we observed no significant indel formation at the target sites. And so these results together show that you can evolve and engineer Cas to enable robust activity in mammalian cells without sacrificing some of these key features of natural type 1 CRISPR-associated transposases. Okay, and then finally, I'll end by talking about our first foray into disease-agnostic therapeutic gene editing. The idea actually came out of a brainstorming session that we have every year in our group offsite meetings. So 3 years ago, my example that I presented to the group of what we call science karaoke was this concept. How can we use one composition of matter, one prime editor, to treat multiple seemingly unrelated genetic diseases that might affect different genes, different pathways, different tissues. And this is the concept that we call disease-agnostic therapeutic editing. And this effort was led by Sarah, Stephen, and Keita in my lab. And I'm really happy that they were willing to take on this project. Okay, so as our first example of disease-agnostic therapeutic editing, we wanted to go after premature stop codons. They are a cause of about 30% of genetic diseases. And if you just look at ClinVar, almost a quarter of ClinVar's 200,000 pathogenic alleles are nonsense mutations that create premature stop codons. Now, there is a strategy that researchers already know of to rescue premature stop codons regardless of where they occur, and that is to use a suppressor tRNA. These are tRNAs in which the anticodon has been changed to complement a stop codon. And while they can rescue nonsense mutations, they've proven challenging to develop into medicines for, I think, a variety of reasons. First, you need to provide them for the lifetime of the patient. And so whether you're dosing them in a transient form, like with a lipid nanoparticle, or providing them with an AAV, which probably will also at some point need redosing, and it's not clear how we can do that, that can be more challenging. In addition, if you just change the anticodon of a suppressor tRNA to complement its stop codon, you usually end up with a fairly modest potency suppressor. And historically, that has required overexpression to get substantial levels of read-through of premature stop codons. And when you overexpress a tRNA, or really any component of the translational machinery, that can cause global perturbations to translation, which can lead to toxicity. So some of the boundary conditions of our idea here was how can we enable suppressor tRNAs to be more effective at treating genetic diseases, in particular without requiring their overexpression, which is a real challenge. If you can do that, suppressor tRNAs are thought to be, if you don't overexpress them, quite well tolerated in eukaryotic cells for a variety of reasons. Cells have basically evolved multiple safeguards to distinguish natural termination codons from premature stop codons. First, there's a difference in their statistical representation. TAG is the most common premature stop codon, but the least common natural stop codon. Second, natural stop codons are usually found in clusters of multiple redundant stop codons, all of which have to be suppressed to lead to read-through past the natural stop codon. Third, and I think very important, release factors are recruited. Release factors terminate translation, and they are recruited to the 3' UTR of messenger RNAs, which is near natural stop codons, but often distal from premature stop codons. And so these release factors more efficiently outcompete suppressor tRNAs that are trying to read through a natural stop codon versus a premature stop codon. And then fourth, there are a variety of pathways that cells have evolved to degrade proteins that end up being translated into their 3' UTRs, like the nonstop protein decay pathway. Genetic data in people supports this idea. You have 418 high-confidence tRNA genes, roughly. And there are quite a few healthy individuals, like 50% of humans, that are missing some of these tRNA genes, or that have mutations, including in the anticodon loop of these tRNAs, that should create miscoding or even suppressor tRNAs, yet these individuals have been documented in some cases, to be healthy. So all of this background led to this idea. So our idea is to take advantage of prime editing search and replace capability to convert a dispensable endogenous tRNA gene into a suppressor tRNA. If this conversion can be done safely and efficaciously, then in theory, a one-time treatment of a single prime editor might provide a therapy for multiple genetic diseases that are all caused by the same flavor of premature stop codon. For example, hundreds of thousands of patients that suffer from premature stop codons that cause cystic fibrosis and Stargardt's disease, phenylketonuria, and Duchenne's muscular dystrophy, in principle, might benefit from a single prime editing drug that unifies patient cohorts both within a disease and even perhaps among different diseases. So to test to develop this idea, we began by using prime editing now as a research tool and 17,000 peg-RNAs to convert each of your 418 endogenous human tRNA genes into all three types of suppressor tRNAs, first just by replacing their anticodons with CUA, UCA, and UUA. We performed this experiment using a lentiviral library, and we assessed nonsense suppression activity by the ability to read through a premature stop codon in GFP. The screen identified several families of tRNAs that can serve as suppressors upon simple anticodon-only replacement. But consistent with the literature and what I told you, even the best of these anticodon-only suppressors, when installed at a native genomic tRNA site, so not just blasting the cell by overexpressing the suppressor tRNA, they showed fairly low potency, supporting up to about 5% of the amount of full-length GFP from a positive control GFP that lacks any premature stop codon. So we wanted to identify improved suppressor tRNAs that could more efficiently suppress premature stop codons, and generated tens of thousands of variants of the best performing suppressors in the last slide, including every possible paired and unpaired nucleotide substitution, every single base pair deletion, as well as many synthetic and natural leaders and terminator sequences. We used that same screen and learned many, many things about the determinants of tRNA function, including the importance of matching endogenous leader sequences with their cognate tRNAs, which is, I think, quite interesting. But in the interest of time, I'll just focus on the discovery of tRNA variants, including the ones shown here, that show a dramatic increase in suppression activity, such that now a single endogenous tRNA, genomic copy converted into the best of these super suppressors, no longer yielded 5% of wild-type GFP levels. They were now getting to up to 35% of wild-type GFP protein levels. And that level of suppression efficiency from a single genomic tRNA gene suggested to us that it might be possible to safely and effectively rescue many loss-of-function genetic diseases caused by nonsense mutations. So once we identified the sequence of a highly active engineered human suppressor tRNA, we next did what we do a lot in our lab. We optimized prime editing systems to efficiently convert an endogenous human tRNA, this happens to be an endogenous redundant human leucine tRNA, into this optimized suppressor. And after exploring a variety of peg-RNA and prime editor choices, we could achieve about 80% prime editing efficiency to install this optimized suppressor tRNA in 6 human cell models of Batten disease, Tay-Sachs disease, and Niemann-Pick type C1 disease arising from premature stop codons. Consistent with many previous studies suggesting that prime editing, by virtue of its mechanism, is inherently resistant to off-target editing, we did not observe any off-target prime editing using 2 different genome-wide off-target detection methods. And now in the 4 human cell models of nonsense mutations that cause Batten's disease and Tay-Sachs disease, when we treat with a single prime editor and peg-RNA, we can restore 20 to 70% of the wild-type activity levels of what used to be the missing enzymes. So these are 20 to 70% of wild-type enzyme activity levels in these two different Batten disease human cell models, which have different premature stop codons, and these two premature stop codons that cause Tay-Sachs disease. Now, for Niemann-Pick disease type C1, that gene is a cholesterol transporter, not an enzyme, so it's not as easy to assay. But we observed substantial rescue, at least of full-length protein production upon treatment with the same prime editor. So this is a cool example because it shows that one prime editing agent, the same prime editor is used in all 6 of these examples, can at least partially rescue 6 premature stop codons in 3 unrelated genes that cause 3 distinct genetic diseases. So what about the safety? We worried a lot about whether this approach would be able to efficiently suppress premature stop codons without perturbing the cell through natural termination codon read-through or other unforeseen effects on the genome or the transcriptome or the proteome. So I showed you off-target gene editing, which appeared to be genome editing, which appeared to be minimal. And to assess the other kinds of effects on the cell, we first wanted to assess how much of this suppressor tRNA are we making when we convert an endogenous genomic copy to our super suppressor. And the answer, somewhat to our surprise, is that our optimized suppressor tRNA is made at only about 40% the normal level of the tRNA, the endogenous leucine tRNA that it replaced. In other words, this optimized suppressor tRNA is being underexpressed, not overexpressed in the genome. Because again, we're not relying on, for example, viral overexpression of the suppressor tRNA. We've just installed it at an endogenous single tRNA gene site using the endogenous promoter and leader to regulate expression levels. And since a known consequence of tRNA overexpression is global perturbation of translation that can be toxic, we were encouraged by the fact that we could achieve potent suppression without overexpression, which we think will be critical for safety. And one of the motivations behind PERT, as we call it, prime editing mediated read-through of premature termination codons, is our hypothesis that with underexpression of a suppressor tRNA, together with the biological safeguards against natural stop codon read-through that I previously summarized, we might actually minimally perturb cells. And so we looked for several kinds of abnormalities in human cells following PERT treatment. We don't observe any transcripts that differ more than 2-fold in abundance before or after PERT-mediated prime editing-mediated suppressor tRNA conversion. We also performed whole proteome mass spec and observed that the only protein that was significantly different in abundance was in fact full-length GFP before and after PERT treatment, because these cells contained our premature stop codon in GFP. And then we looked for peptides explicitly that correspond to read-through of the 4,036 natural human proteins terminated with a TAG stop codon. So you can predict what peptides should be generated if that TAG stop codon is read through, and you can then perform mass spec looking globally for the presence of these read-through peptides. And while we did observe lots of peptides corresponding to read-through of the premature termination codon in GFP, we did not observe any peptides, somewhat to my surprise, for any other protein corresponding to translation past the natural TAG stop codon. We increased the sensitivity of this experiment by picking the 69 most abundant proteins that use a TAG stop codon and performing target ion peptide mass spec of the predicted peptides from natural TAG stop codon read-through. And once again, the only peptides that we observed at a statistically significant level were upon PERT treatment were those corresponding to read-through of the premature GFP stop codon. So these data suggest that PERT does not induce significant read-through of natural termination codons under the tested conditions, likely due to the mechanisms by which cells minimize the frequency of natural termination codon read-through that I already summarized, and the short lifetime of any proteins that get read through. And then finally, we moved the PERT system in vivo. We were disappointed to quickly learn that mouse tRNA genes are different than human tRNA genes, so we had to repeat more or less everything I presented for the mouse tRNA genes. And then we injected AAVs encoding a prime editor targeting a mouse tRNA, converting it into optimized mouse suppressor, first into mice with a premature stop codon in GFP. And this resulted in rescue of about 25% of wild-type GFP levels among the transduced tissues, in this case in the brain. And then we used the same prime editor, literally the same AAV, to inject a mouse model of Hurler syndrome. This is a severe lysosomal storage disease caused by a premature stop codon in a gene called IDUA. And here we observed about 7% editing and about the same amount of IDUA enzyme activity compared to wild-type mice. And as is the case with many loss-of-function genetic diseases, this amount of wild-type IDUA enzyme should be sufficient to rescue Hurler syndrome. And indeed, by a variety of histological and phenotypic analyses on multiple tissues, we observed complete or near complete rescue of Hurler syndrome pathology on these mice, including foam cell accumulation, vacuolization, and the accumulation of glycosaminoglycans. Okay, so sorry for the AV issues. I'm not sure what happened, but to summarize the talk, I've shown you how we can use base editing and prime editing to enable mutation-specific therapeutic gene correction. This has occurred in at least 23 clinical trials with at least 9 positive clinical readouts and a number of positive patient outcomes. You can perform targeted gene size integration in human cells. I showed you 2 ways, either using a prime editor and a recombinase or using an evolved CRISPR-associated transposase. And we're hopeful that these types of technologies will enable mutation-agnostic therapeutic editing, where you can put an entire healthy copy of a gene, but critically at a location where you have a high confidence it will be able to perform its endogenous function without causing toxic overexpression or misregulation. And finally, I showed you an example of disease-agnostic therapeutic editing, where we can use prime editing to convert endogenous tRNAs into high-potency engineered suppressor tRNAs so that a single endogenous copy can rescue nonsense mutations in human cells or even in mice with just the same composition of matter. And finally, that leads me to thank this incredibly talented group of graduate students and postdocs, as well as collaborators shown in yellow. Many of the projects shown here were real team efforts involving multiple labs. The leads I've marked with a star, and I'm very grateful especially to Mark Osborne's lab on the PERT work, Sam Sternberg's lab for EvoCast, and Mark Osborne, Elliot Chaikov, Torsten Meisner, and Jakob Tolar on the EvoPassage work. And a variety of labs have contributed to the development of base editing and prime editing and their clinical applications summarized here. And then finally, thank you very much for your attention. And if there's any time, I'm happy to take some questions.
Thank you so much, David, for a wonderful presentation. There are plenty of questions, so we have 2 minutes until 2, and if you could stay a little longer, we can take some of these questions.
Yeah, I'll do my best. I'll turn on my camera. Hopefully you can see me and I'm not sharing anymore, I think.
Yeah, this works.
Okay. So yeah,
awesome.
Go ahead.
So I'll start taking some questions from the Q&A. One question says, how would you envision delivering your PERT in vivo, particularly if passing the blood-brain barrier is important?
Yeah, so we are working on doing therapeutic gene editing in the central nervous system. And one of the priorities of this new Center for Genetic Surgery, CGS, that we've set up at the Broad is actually to purposefully pursue what we think is one of the big frontiers remaining in therapeutic gene editing, namely editing in the brain. If I had to bet on just one or place one bet on one type of delivery mechanism, I'm actually excited about these blood-brain barrier crossing AAVs. like CapX, Apertura, Apertura's AAV, Ben Deverman's lab has developed some of these key concepts. And I think that may be in the short and medium and maybe even long term a good solution for delivering these types of gene editing agents into the brain. There are other efforts underway, of course. People are trying to develop LNPs that can access the brain, peptide nanoparticles, and virus-like particles. But I think the first clinical readouts will probably come from these blood-brain barrier crossing AAVs. And so I'm excited by that. There are also, of course, ways to directly inject into the central nervous system. But I think maximizing patient accessibility and minimizing patient adverse events would benefit from being able to administer a systemic vehicle that could cross the blood-brain barrier. And so, hopefully the ongoing work that's about to enter the clinic on these blood-brain barrier crossing AAVs will provide us some encouraging news.
Thank you, David. Another question here, have you considered ways to target the engineered endogenous suppressor tRNA to a specific transcript? This could help further mitigate safety concerns with perturbation of off-target translation while also improving efficiency of on-target transcript read-through.
Yeah, it's an interesting idea. We haven't done any experiments there, although we've contemplated various ways you can add on to regions of the tRNA that show some tolerance for addition to allow aptamer-like binding of the tRNA to various targets. I'll say that there's a trade-off. One of the reasons you would want to do this as opposed to just correct the mutation in the patient, which on paper seems like a more direct way to treat a disease, is because of that disease-agnostic approach, that you might imagine having maybe a dozen flavors of PERT agents that cover a polar, a hydrophobic, and a charged amino acid for all three stop codons. If you had a freezer full of these variants, then in my long-term dream, you might be able to pull a variant out of the freezer when you diagnose a patient that has a serious genetic disease caused by a premature stop codon. Sequencing is now very fast and very economical. And once you identify that, okay, this is a likely pathogenic variant premature stop codon caused by, the mutation of a leucine to a premature stop codon, then perhaps you can pull out an existing syringe of stuff and actually treat the patient. That disease-agnostic ability, of course, goes away if you are targeting the specific suppressor to a specific gene or to a specific gene variant. But I think it's a cool scientific idea, and if you could just add to the specificity of engineered variants of translation by recruiting the components to a specific transcript, that would certainly be a really cool way to manipulate biology.
That sounds super cool, David. All right, another question here. What is the best way to correct large structural variants, such as hundreds of excess triplet repeats that have other effects, such as hypermethylation?
Yeah, we have a manuscript in review now on prime editing triplet repeat disease loci to remove large hundreds of CAG or GA repeats, for example. And you can do that. It's a more challenging than normal kind of edit to make, but prime editing does have that capability to directly and precisely remove the offending pathogenic repeat stretch. So hopefully that work will come out this year. I would be very disappointed if it weren't published in 2026. In terms of other kinds of large-scale pathogenic variants, we still, I think, humans don't have the ability to correct, mega-base deletions with any kind of therapeutic efficiency and practicality. So there's, and translocations, while you can imagine using prime editing in a quad flap way to make or correct translocations, and there's been some research and some literature reports along those lines. I'm not aware of efforts to really advance that kind of work into the clinic. So there are still important areas of editing space where I think we need additional work.
Thank you, David. Maybe we'll take maybe one or two more questions, if that's okay with you.
Sure.
So is editing the PTC back to wild-type codons a superior treatment than using suppressor tRNAs? A lot of patients share similar nonsense mutations.
Sorry, can you read the question again? It cut out at the end.
Is editing the PTC back to wild-type codons a superior treatment than using suppressor tRNAs?
Yeah. So I think it's the direct correction of the premature stop codon offers a lot of benefits over a suppressor tRNA approach, but of course you have to make a custom editing agent for each premature stop codon. That's the point. So yes, if you have the good fortune of having the organization and the in-kind contributions and frankly the serendipity and the good luck and the talent of the team, the multiple teams that generated baby KJ's base editor, then yes, you can do that. But I don't think replicating baby KJ 20 times is going to be the answer for how to best treat lots and lots of patients. If we want to have that kind of success on a much, much larger scale of hundreds or thousands or even millions of patients, we're going to need approaches that allow one medicine to address more than one mutation. And that's sort of the underlying theme of this talk, is you can use precise gene editing in creative ways to enable therapies that have the potential at least to treat lots of different patient populations.
And I'll ask a question, David. I can see that there isn't too many off-target effects when you're using the suppressor tRNA, but as you may know, these premature stop codons are important in the cell as checkpoints. So I'm trying to understand, like, how do you ensure that when you use the suppressor tRNAs in the future in humans, you're for sure not affecting any other, circumstances where these premature stop codons are important to be there and not suppressed.
Yeah, I think it is because it's a sort of new approach to do it by gene editing. It will be important to do a comprehensive safety analysis. Even though we did, I think, what would be considered on an academic scale fairly comprehensive, it's certainly not what I would consider comprehensive on an IND enabling scale. So I think we would look for specific, both global changes like we did with global transcriptomics and proteomic changes, but also look, those global methods tend to trade off coverage for sensitivity, just like whole genome sequencing. You're not going to see a 0.1% variant with whole genome sequencing. And I think you have to complement that ideally with targeted investigations. So if there's a premature stop codon, an endogenous premature stop codon that one is concerned about because of its role in regulating X, take a look at, what fraction of the time do you observe read-through and downstream signaling as a result of PERT? Again, I think a lot of the potential downsides are mitigated by the fact that if you keep the stoichiometry of translation intact, and especially if you underexpress, not overexpress the suppressor tRNA, it may not be that much of a perturbation. That's, I think, the best explanation for why, again, somewhat to our surprise, we did not observe mass spec evidence of global read-through. I thought of the 4,000 naturally TAG terminated protein coding sequences, we would observe at least, dozens, if not hundreds, that were read through at least a little bit. We also have to keep some risk-benefit perspective here, which I think is, FDA's opening its eyes to, that if you are in baby KJ's state and you have a 50% chance of dying in infancy, maybe you worry less about read-through of natural premature stop codons if you know that the prime editor in your freezer could be immediately deployed to minimize the progressive brain damage that the baby would otherwise suffer from accumulation of ammonia in the blood. So I think there are a lot of serious genetic diseases that unfortunately are serious enough and lack an effective treatment that you could make a risk-benefit case for trying something like this if we can, clear a reasonable IND-enabling study array, which we're trying to advance PERT, both covering other kinds of stop codons like arginine to stop, which is a UGA stop, is a very common kind of, all those R265X, R270X kind of mutations are all arginine to UGA stop codons. We'd love to have a flavor of PERT to do that, which we're working on now. So there's still a lot of work to be done, and I think we will have to keep an eye on those pathways like what you described, even though thankfully it looks like the vast majority of genes that are just read and terminated the old-fashioned way don't appear to be perturbed at a strong level by a single endogenous installation of a suppressor tRNA.
That's awesome to hear, David. Super cool stuff. All right, we're 11 minutes past time. So thank you so much, David, for a wonderful presentation and, staying longer to take all these questions.
My pleasure.
It was, fantastic to host you here, and hopefully we can bring you to NCAT sometime soon. And everyone else, thank you for being here, and we'll see you next quarter with another awesome webinar. All right, have a good one, everyone.
Thank you. Bye-bye.
Yeah, bye.